Hello everybody, and welcome back to Creation Myths. I have a new creation myth for you today, and it's a big one. It's the waiting time problem. So what in the world is the waiting time problem? This is basically the idea that creationists argue that it takes too long to get new stuff. Somehow. New traits, new mutations, whatever. Generally, we're talking about combinations of mutations or new genetic traits. So, say you need 8 mutations to do a specific thing compared to whatever hypothetical ancestral state. Creationists will argue that, well, it would take too long to get those 8 specific mutations, therefore that thing could never evolve. We see this written and talked about a lot. So here we have a short article in Creation magazine on it. Here we have Dr. Rob Carter talking about it and how it's a huge problem for evolution. We'll see if that's the case. Spoiler, it's not. And really notable that I think for the waiting time problem is we have actual peer-reviewed creationist publications on this topic. Now, they're not in, like, the greatest journals. They're kind of in, like, those theoretical biology journals, which are, but credit where it's due, creationists are actually publishing real peer-reviewed work on the waiting time problem. So here we have John Sanford and his co-authors in Theoretical Biology and Medical Modeling from 2015, The Waiting Time Problem in a Model Hominin Population. And then more recently in the Journal of Theoretical Biology, we have Oster, Beckley, and Gager on the waiting time until coordinated mutations get fixed in regulatory sequences. So they are actually doing, like, real work on this, as opposed to just like Nathaniel Jeanson writing more books on his made-up human genetic stuff, right? They are actually doing peer-reviewed publications on this specific topic. So why are we seeing this work on this specific issue? Well, the waiting time problem is one answer to what I like to call the what's the barrier question. This is like a common question that people will ask creationists because all creationists accept some degree of evolutionary change, some more than others, right? Some creationists will say, well, I accept microevolution, but not macroevolution. And some creationist organizations actually accept a lot of evolution, and they cram it into a very short time frame immediately after Noah's Ark, right? You've got like this hyperspeciation following Noah's flood. So a question that often comes up directed at creationists is, okay, you accept that A and B and C have a common ancestor. Well, what about D, E, F, G, blah, 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 all the way down? What's the thing that if the mechanisms work to get from a common ancestor to extant species at one scale, what prevents those processes from operating at larger scales? And creationists have come up with a few different answers to that question. One answer is that, well, evolution is mostly going to degenerate. So this is like Michael Behe's Darwin devolves hypothesis or John Sanford's genetic entropy hypothesis, right? Evolution is mostly going to degenerate, so you can't generate new things that you need to get that degree of evolutionary change. Another proposed answer is that if you have characteristics that meet a set of criteria, then those things are not going to be able to evolve. So think about like irreducible complexity from Dr. Michael Behe. If something meets the criteria for irreducible complexity, then it supposedly cannot evolve. Now, I've talked about that before, and that's not true, but that's the explanation that creationists will provide. Well, the waiting time problem is another attempt to answer that question. Instead of providing a specific set of criteria for a trait that can't evolve, they're just saying some number of genetic changes over some amount of time is too many in order for a specific trait to evolve in whatever context you're talking about. And there are a number of different contexts where this argument applies. So that's where this sits kind of in the broader pantheon of creationist arguments. It's an attempt to provide an answer to the what's the barrier, what's the mechanism that prevents evolutionary processes from operating over some large scale. There are a couple of different ways you see this argument applied. One is to talk about specific features and say, well, if you need more than a certain number of genetic changes, then it would take too long for that feature to evolve. In fact, we can go to Dr. John Sanford saying that if you exceed so many genetic changes or genetic characteristics that are required for a specific trait, it would take longer than the time since the Big Bang to evolve.
8 mutations, waiting for 8 specific mutations takes more time than the time since the Big Bang.
Another approach that creationists take is to say that a specific speciation event would take too long. Basically, if you have two species that biologists claim are related, which they are, because all species are related, but if you have two specific species, you say they shared a common ancestor at some point in the past, creationists will say, well, there's way too many differences between those two since their most recent common ancestor for evolution to have generated all of that level of divergence. Therefore, they could not have evolved from that common ancestor in the time frame that evolutionists claim, right? That's another way this argument is made, for example.
Even granting a best-case scenario for evolution by generously assuming human and chimp DNA is 99% identical, the remaining 1% would still be a difference of 30 million DNA letters, an impossible genetic barrier for evolution to traverse in 6 million years.
So why are creationists wrong when they make these claims? Well, let's find out. I'm going to group these kind of objections into two categories. The first being just like general problems. Like basically, creationists are operating under a misunderstanding or a misrepresentation of evolutionary theory in order to make this argument. So the first thing that creationists do when they talk about the waiting time problem is they treat evolution as a serial rather than a parallel process. They say, in effect, you have to have one mutation occur within a population, and then the next mutation occurs, and then the next one, and you have to get each required mutation in sequence within that population in order for the ultimate, the trait at the end of it to appear. That's wrong, because evolution doesn't work that way. Evolution is what you call a parallel process, where you have lots of individuals within a population evolving at the same time, in parallel with each other. So if you look at this simple representation, we have generation 1, generation 10, where each color for a circle represents a specific mutation or a specific genotype. So each circle is an individual, they all start off the same in generation 1, and then mutations occur, and they go away, and different mutations occur. So by the time you get to generation 10, you have a bunch of different mutations present in this population. So it's not that you have to have one mutation in the population, then the next one, then the next one, then the next one. You can have the population sampling many different mutations at the same time. That's how evolution actually works, as a parallel process as populations evolve over generations. Related to this, creationists exclude recombination from the discussion when they talk about the waiting time problem. And this is just a little excerpt from that Hofstra et al. 2021 paper. This came out just in, I think it was August of 2021. They're talking about getting a specific regulatory sequence within a population, and they say it is reasonable then to believe that recombinations between two parental regulatory sequences can be ignored. No, that's completely unreasonable. That's completely unreasonable. Recombination happens for most organisms during sexual reproduction. Sexual reproduction is costly compared to asexual reproduction. It typically takes longer, it requires more energy, you have to find a mate, you have to attract the mate, you have to actually mate, it can be dangerous, it's a vector for spreading disease. There are a lot of costs to sexual reproduction. But the reason it tends to be beneficial anyway is that it allows you to mix and match genotypes between different individuals. So looking at our hypothetical population of circles over here, if you have reproduction between the lineage that has the blue mutation and the lineage that has the pink mutation, well, their offspring could end up with both and then do recombination in their germline to end up with a gamete that has both the blue mutation and the pink mutation found together. So you don't have to experience one mutation at a time. You have populations that have many mutations present simultaneously, and then through sexual reproduction and the accompanying recombination, you generate combinations of those mutations faster than if they had to accumulate one at a time in sequence. So when you're considering the waiting time problem, you absolutely cannot exclude recombination. The third and final kind of conceptual error that creationists make here is that they assume a specific target. And we could see this again in the 2021 paper I previously referenced, and also in a paper by Douglas Axe from 2004. In the 2021 piece, they say, fifth, we have analyzed the waiting time until a fixed and pre-specified target is reached. And back in 2004, Douglas Axe, in a kind of not exactly a waiting time problem paper, but along those lines. He said, talking about the likelihood of generating specific protein folds, he said it's likely that the sample space can be restricted to sequences carrying the hydropathic signature of a known fold. Axe also writes that the difficulty of specifying a working beta-lactamase domain is assessed here. So in both of these cases, they're talking about having a specific target sequence that they're working towards, and they're calculating how long it would take to arrive at this specific target sequence. But that's not how evolution works. There are lots of different sequences that will work approximately equally well to accomplish a particular function. And we could see this in the peer-reviewed literature. For example, there was a study from 2008 finding that random sequences rapidly evolve into de novo promoters. This is a really interesting study. What the authors did here is they took a regulatory sequence within the E. coli genome that works out to about 100 nucleotides, and they replaced it with random strings of 100 nucleotides. And what they found was really interesting. They found that you don't need those specific 100 nucleotides that we typically find in E. coli. It turns out a lot of strings of 100 nucleotides will accomplish the same function. So here is a figure from this paper. The specific thing that they knocked out is called the promoter for the lac operon. It's basically the part of the regulatory network that allows the genes to be expressed. And we don't need to go into more detail than that. So in this figure, we have on top, we're looking at the expression levels of those lac operon genes with just the random 100 nucleotides. And then down here in the bottom, we have the random 100 nucleotides, where each one has experienced one mutation. We have the fraction on the y-axis for both cases, and in the x-axis, we have expression relative to the WT that's wild type, that's the normal lac promoter. So here, this line is the normal lac promoter. So what we see is that just random strings of 100 nucleotides, you get some lac expression right off the bat, no mutations required. With a single mutation to each of these sequences, most of them, the vast majority, will exhibit some lac operon activity, meaning they will work with a single mutation. So you take random 100 nucleotides, and then a single mutation, and you end up with functional sequences. The studies that creationists do that set a pre-specified target, they're just looking for the equivalent for this study is the probability of re-evolving the wild type lac promoter. But that's not the question in evolution. The question is, what's the probability of getting something that will work as a lac promoter? And it turns out that's really easy to do. So setting a pre-specified target is completely the wrong approach when you're trying to evaluate the likelihood that a specific trait or a specific function could evolve, because we now know that there are lots of different ways to accomplish the same thing when it comes to like biochemical functions and genetic sequences. So those are the general problems with the waiting time problem as argued by creationists, and those alone are enough to invalidate the concept. But let's look at a specific example that they like to apply this problem to. Let's look at human evolution. The creationist claim here is that there are too many differences between the human and chimp genomes for the 6 to 10 million years of evolution since we diverged from our most recent common ancestor.
A difference of 30 million DNA letters, an impossible genetic barrier for evolution to traverse in 6 million years.
But even if to get 1% of the genome different requires 30 million mutations, that's a lot of new information.
So let's run the math on these creationist claims. The first problem is that there are two lineages from the human-chimp most recent common ancestor to extant humans and extant chimps. So take a look at this little diagram here in purple. We have our most recent common ancestor, the thing that we evolved from. And then on the bottom, in red, we have humans, and in blue, we have chimps, and creationists say, well, there's 30 million differences or 300 million differences. Let's take 30 million, given their lowest estimate, and say there are 30 million differences between us and chimps. So creationists say you need to get 30 million differences along that human lineage, and that's just too many mutations and not enough time. However, that's wrong because chimps are also evolving too. We didn't evolve from a modern chimp. That hasn't changed in 6 to 10 million years. So Instead of 30 million differences on the human lineage and 0 on the chimp side, it actually, if you take that 30 million number as valid, it should be about 15 million on one side and about 15 million on the other side if you assume it's about 50-50. It's actually more on the chimp side, but we're not even going to worry about that. We're just going to give them the benefit of the doubt and say it should be 50-50. So right off the bat, the creationist number is 2 times too high. The second problem is that creationists assume that all of those differences are beneficial. They say, oh, well, you need 30 million new beneficial mutations to get from the most recent common ancestor to the modern human. That's wrong. Most of those mutations aren't doing much of anything, and we know this because we can detect positive selection. Positive selection is when something beneficial appears in the genome, and then it is under selection that basically it increases in frequency because the individuals with that new beneficial trait have more offspring, so they make more genetic contributions in subsequent generations, so it increases in frequency. So we can detect that. We can find the signatures of positive selection in the human genome and actually figure out how many new beneficial alleles there are in the human lineage compared to the chimp lineage. And when we do that, when we do those surveys, we find out that it's about 1,000. So I'll show you two studies here that have arrived at slightly different answers, but they're similar enough. So this first one from 2006, they looked at about 13,000 genes, and they found that there were just under 500 human genes that they say deviated from the strict molecular clock, meaning they were under selection of some kind. Now they specifically found, in a more stringent measure, they specifically found 108 genes in humans that were under positive selection. But we're going to be nice to creationists, and we're going to use the bigger number, and we're going to say about 500. Now, they only surveyed 13,000 genes, and there's about 20,000 genes in the human genome, so let's just, again, be nice and just double that number to about 1,000, right? About 1,000. Now, another study, 2007, they actually identified 154 genes that were under positive selection in the human lineage, which is kind of in line with the 108 over here. It's a little lower than the 469, but like it's in the same ballpark, right? Now, this study also, the second one also only looked at about 13,000 genes, so it's an underestimate. But again, it's lower than this previous study. So even if we're nice and we double it to, give the creationists the benefit of the doubt here, we're still looking at about 1,000 new fixed alleles in the human genome compared to the chimp lineage. So the creationist calculations, such as they are based on just made-up wrong numbers. It's not 300 million differences. It's not 30 million differences. The actual number of new beneficial alleles within the human genome is in the literature, and creationists can find it if they want to look for it, and it's about 1,000. That's kind of a problem, right? Let's see just how big a problem it is. As we just said, the actual data show about 1,000 new beneficial alleles in humans versus chimps. But you know what? These studies, they were from 2006 and 2007. They're imperfect. Even modern techniques are imperfect for detecting this kind of thing. So let's just double that number in case we missed any. Let's just make it 2,000. Just say 2,000. And also, you know what? This was only looking at protein coding genes. Let's throw in another 1,000 for regulatory sequences. We had our estimate of 1,000 just for genes. Let's just say also 1,000 for regulatory sequences. So we'll just throw in another 1,000 because we're feeling nice. There's probably some mutations in regulatory sequences that are new and beneficial in the human lineage. I bet that's a thing. But regulatory sequences are a much smaller fraction of the genome than protein coding genes. So if there are 1,000 new beneficial alleles in protein coding genes, are there going to be 1,000 changes in regulatory sequences that are new and beneficial? No, it's going to be a lot less than that. But let's be nice and let's just make it 1,000. So now we're up to 3,000 differences. 3,000. And you know what? Let's multiply by 10. Let's just give them an order of magnitude for no reason other than, why not? Let's say, what if, we're talking about alleles here, right? We're looking at alleles under selection. An allele is not a mutation. An allele is a sequence. So maybe there's a lot of mutations in those alleles. There's more than one. Maybe there's more than 5. Let's multiply by 10, right? In case it's 10 mutations per allele that are new and beneficial in the human lineage. That gets us a grand total of 30,000 new beneficial genetic traits in humans that had to appear and be selected for since we diverged with our most recent common ancestor with chimps. 30,000. Well, here's the fun part. That is 3 orders of magnitude lower than the lowest creationist claim of 30 million. The range that creationists gave for how many new beneficial mutations you need within the human lineage was between 30 million and 300 to 400 million, right? That's the range they gave. The actual number, if you use completely bonkers assumptions in the creationist favor, including multiplying your empirically determined number by 10 for no reason at all other than you're feeling nice to creationists, you get 30,000. That's 1,000 times lower. Not 1,000 lower, 1,000 times lower. A factor of 1,000 below the number that creationists claim. So is there any merit to the creationist claim that there is a waiting time problem for human evolution? No, it's completely baseless. There is no validity to that claim at all. To summarize, creationists claim the so-called waiting time problem shows how evolutionary processes can't explain extant biodiversity. However, their argument involves multiple conceptual errors, such as assuming serial rather than parallel processes, disallowing recombination, and having a specific target pre-specified, which alone invalidate their argument. And the specific applications of this argument, such as human evolution and our divergence from our most recent common ancestor with chimps, involve significant methodological errors and failures to review the relevant literature. The actual data show a very small number of new beneficial traits that must evolve in that specific case of human evolution, several orders of magnitude fewer than what creationists claim. So, in conclusion, there is no waiting time problem in general or in the specific case of human evolution. So the so-called waiting time problem, that is, as always, a creation myth. Thank you for watching. Please like, subscribe, share, and don't get fooled.